All right, so we just finished up with the lytic cycle. So in this cycle, we had the virus kind of taking over the host cell and having the host cell replicate lots of new bacteriophages and releasing those through the process of rupturing or lysing the host cell. There's another cycle, another way that bacteriophages are capable of replicating, and that's called the lysogenic cycle. So these bacteriophages are typically called temperate phages, and they can kind of go between the lytic cycle and the lysogenic pathway. So the first two steps of the lysogenic cycle is kind of that attachment and the penetration, kind of same as the lytic cycle pathway. But following penetration, the phage genome is incorporated into the host cell's genome, and it forms what we call a prophage, not a provirus, but a prophage. So in this case, we are going to insert the bacterial's genome, but at some point we can take it back out again. All right. So as the infected bacterial cell divides, it's going to copy its genome, and its genome now includes the prophage or the virus's genome. So therefore, from a single infection event, this can ultimately result in many cells carrying the bacteriophage's genome. And if the host cell carrying the prophage becomes stressed, the prophage may kind of cut itself out from the host genome and enter the lytic replication pathway. So it's kind of like it's abandoning ship, right? So if it kind of gets some chemical cues that the bacteria may be on its way out, may be dying, it allows the bacteria, or the bacteriophage, to find a new host, right? So it kind of ditches its current host and goes out and tries to find a new one. So this, that we're going back to our image here that we had of the lytic cycle, but it's showing the lysogenic state right here, not really fully showing it, but if you can think about the bacteriophage, right, has adhered to the outside, it's injecting its DNA into our bacteria, and now that little line of genetic information there is incorporated into the circular strand of DNA from the bacteria. So this is a blow up of what is occurring. In pink, we have the bacterial genome, and in blue, we have the bacteriophage's genome that is being inserted into it. So every time this bacterial cell then divides, it's going to replicate its DNA. And when it replicates its DNA, it's going to replicate that little section right there, which is the bacteriophage's genome. So that will then be inserted into the new bacterial cell that's produced. And every time the bacteria replicates itself, it's going to replicate that viral genome. So as it was saying in the previous slide, it's kind of this silent interaction, this kind of silent method of replication. So it doesn't kill the cell, right? But it's replicating kind of alongside of the bacterial cell, or I shouldn't say alongside, it's actually inside, but I meant kind of in tandem with the bacterial cell. Now, we said at some point, right, so here on the next slide, oops, got to exit drawing there, it talks about induction. So it says the prophage becomes activated and enters the lytic cycle. So at some point, it gets some kind of cue, not 100% sure of what this is. We don't always know what the case is, but it gets some type of environmental signal, some environmental factor. So it could be UV radiation, could be some type of epigenetic factor, but it gets some kind of cue that says, okay, it's time to stop this type of replication and let's go into the lytic cycle. So at that point, the prophage pops out of the bacterial genome, and then it will start producing new viruses. All right, so new capsids replicating just the viral genome at this point, assembling them into actual viruses, and then it will lyse the cell. Right? So in the lysogenic state, we're not producing whole viruses at all. There's no capsids that are being made, right? It's only in the lytic cycle that we actually produce these whole new virions that are produced, okay? But a temperate phage can kind of bounce back and forth between these two different modes of replication. All right, so prophages, right, which occur during the lysogenic replication cycle, are medically important. And that's because they can confer new pathogenic properties to bacterial cells. And this is a situation that we call phage conversion or lysogenic conversion. So if you think about it, right, since the virus is adding new genes to the genome of the bacteria with that prophage during the lysogenic cycle, it can add new traits as well. So as it's incorporating its viral genome, it may confer some new characteristics to that bacterial cell because the bacteria is going to read the genes and it could read the viral genes. So usually the new genes typically they tend to affect the virulence of the bacterial cell. And so what's added a lot of times is the bacteria has new abilities to produce toxins, or they're better able to adhere to a surface. These are all things that enable the bacteria to survive better. And so let's say, for instance, we've got up there the botulinum toxin. So botulinum toxin is made by bacteria that's called Clostridium botulinum. But the ability to make that botulinum toxin is due to the fact that the bacteria has a gene for the toxin, but that gene was originally a prophage, right? So it received it from a bacteriophage. So that's one instance where lysogenic conversion contributes kind of that new characteristic or that new trait to the bacteria, and that's coming from the bacteriophage during this lysogenic state, right? Now, bacteria can also have multiple prophages within them. So it's not like they may only have one prophage that has been inserted. They could have multiple prophages from a variety of different bacteriophages. So each of the prophages could encode a different virulence factor. And all of those factors together kind of help to make the bacteria much more resilient, right? So that's why this is kind of importance medically, right? Because there are certain properties that the viruses pass on to the bacteria. All right, so